Hello, I'm Alexandra Agiv from Vadim Axelrod's lab at Bar-Ilan University, and today I'm going to present our intracranial study of humor processing, in which we used a 3 minutes long fragment of the movie Circus with Charlie Chaplin. So first of all, we had a unique opportunity to use stereotactic EEG, which allowed us to investigate activity over various frequency bands. And in this study, we examined the neural activity that corresponded to the most funny and the least funny segments of the movie clip we used. So in order to establish these segments, we first conducted a behavioral experiment in which 21 healthy participants watched the movie fragment and then continuously rated it based on its humorousness. We averaged these ratings and we were able to obtain 6 of the most funny segments and 6 of the least funny segments. In the figure below, you can see an example of 4 such segments. Then we moved on to the intracranial experiment, where we had 12 patients who underwent depth electrode implementation at the Hospital of Pitié-Salpêtrière in Paris. And we were able to record LFP activity from 570 contacts in total. You can see in the figure below the distribution of those contacts. And moving on to the data analysis part, we filtered these data into 4 frequency bands: alpha, beta, gamma, and high gamma. And for each one of these frequency bands, we compared the activity during the most funny segments versus the least funny segments, and we focused on the contacts that showed modulation. We were able to observe that the neural modulation occurred mostly in the alpha and the high gamma bands, such that during funny content, the high gamma band showed activation and the alpha band showed deactivation. Thus, we were able to identify 3 types of regions. So we had regions that showed mostly high gamma activation, such as the anterior temporal lobe, which you can see in figure C, and these contacts are marked in yellow. And we had regions that showed mostly alpha deactivation, such as the superior temporal sulcus, and these contacts are marked in black. And we had regions that had both types of activity, such as the posterior IFG, for example. And in the figure below, you can see the amount of contacts showing each type of activity per AAL region. So to sum, we observed that humor processing might be mediated by an interaction between these 2 frequency bands, and we hope our study helps to further understand the functional mechanisms of humor processing.